Very good morning, ladies and gentlemen. I'm very much delighted to see each one of you on the launch of lecture series organized by Academy of Microbiological Sciences and Association of Microbiologists of India. We have been trying for the last couple of years to start this lecture series, and today the dream is coming true. It's all because of your everybody's participation, everybody's say the contribution, and again the credit goes to Professor Gunashekaranji because Academy Council constituted a committee under chairmanship of Professor Gunashekaran, and he could really submit within time the planning and its whole architecture. And now with the insistence of, say, Academy's Council and Executive Council of AMI, uh, the complete list is going to be, I think, completed within next week, and which will be posted on our AMI website as well as in the AMI Life Member Group and other groups, so that everybody will come to know the participants, our say the experts for the lecture series for the year 2026. And one very important decision has been taken that every fortnight there will be a lecture by young scientists as well as the established distinguished scientists. And in that series, today, this is the first day when we are launching the lecture series program, and your presence makes it a wonderful day. I would say that it is a red letter day in the history of Academy of Microbiological Sciences and Association of Microbiologists of India, that we are starting a series, and
हेलो
say the beginning is
मैम वो लाइव
by
भेज दिया मैम
our
लाइव कर दिया अभी ग्रुप
distinguished,
में सेंड ही करा था
say,
मैसेज अनिल
professor,
सर को भी भेज
researcher,
करता
administrator,
हूँ अभी करता हूँ अभी करता
Professor P. Gunashekaran, who
हूँ ओके
is Vice Chancellor at
ओके
VIT Bhopal University, Sihor, and he has served as Pro Vice Chancellor, VIT University, Chennai, Vice Chancellor, Thiruvalluvapuram University, Indore, and Emeritus Professor in Madurai Kamaraj University, Madurai. Dear friends, he has 33 years of teaching and research experience in microbiology, biotechnology, and genomics. Professor Gunashekaran has made significant contribution in the field of agricultural microbiology and industrial microbiology, especially on ethanol fermentation technology and microbial enzyme technology. His research has been focused on Gymomonas mobilis, an ethanol producing bacterium. In addition to the research on Gymomonas mobilis, he has made significant
निशांत एक
contribution
लिंक भेजी यूट्यूब
in
की चेक
process
करियो स्ट्रीम
optimization
ओके चल
and
रही है
production of several industrial enzymes from different bacterial and fungal sources. Dear friends, he has been a researcher of international repute. He visited worldwide,
ऑडियो ऑडियो प्रॉपर सुन
including
के देखो
USA, UK, France, Mexico, Japan, South Korea, Hungary, to conduct various collaborative research programs. Dear friends, you'll be happy to know that he has guided 44 PhD students and published over 180 research articles in national and international journals with a cumulative impact factor of 400, and his research articles has been cited 10,018 times in various journal articles. That shows his credibility in research. As an academician, we know him very well. He has completed various projects from different reputed national and international funding agencies. He has also received several medals, awards, and honors in
सही
India
है
and abroad for his
बस
immense
एक बार
research
सुन
contributions. Just to list few of them, major recognitions include Lifetime
ये फोन
Achievement Award,
एक यहाँ से
Biotechnology
रखा नहीं
Research
मिलता इसको,
Society India,
क्योंकि मैंने ना
Lifetime
विंडो लाइन
Microbiology
नहीं करी, पूरी
Devotion
पूरी
Award,
स्क्रीन देखो इधर
Microbiology
उधर का
Society
सारा
of
दिखता है।
India, Outstanding Service Award, International Board of the American Society of Microbiology, one-time grant award by UGC, Algappa Centenary Excellence Award for Pune and Applied Sciences. There are, I think, many such awards. It is very long, and administratively also he is well known for his contribution at Madurai Kamaraj University and couple of other universities where he has served as vice chancellor. Dear friends, with your permission, now I would like to invite Professor Gunashekaran for the lecture, and this will be a first lecture launch of our lecture series, and he'll be delivering his speech today on metagenomics approach for the development of novel antimicrobial peptides. Dear friends, he will highlight the role of metagenomics in discovering novel antimicrobial peptides from diverse and uncultured microbial communities to combat antimicrobial resistance. We know that which has become a maniac, say at present. The session will also provide a brief overview of key techniques, recent advancements, and future prospects in metagenomics based antimicrobial research. I ask to all the participants because there is a, I think, huge participation uh, on the launch of this lecture series. My urge will be to each one of you to mute yourself so that there won't be any disturbance, say, during the lecture. And if at all, as you have been advised, because every lecture say should follow certain etiquettes, and we are all say the student of science, and we need to follow so that we don't say cause any disturbance during the lecture to Professor. And please follow what when I'm urging, when I'm requesting each one of you, please take care of say it, and we need to give say I think or present an example that when Academy Microbiological Sciences and the Association Microbiologists of India having any program and participants, they are our students, say researchers and teachers, that it's a unique kind of the organization, and we are the soldier of say the AMI and
आवाज आवाज कम
Academy.
कर दे बिल्कुल, बस
I'm sure
चलने दे।
you will ensure me that there won't be any disturbance. And if at all there is, please try to take care of that. Thank you. Thank you very much. And I again wish you a happy new year to you and your family. All the best. Now I invite, with your permission, Professor Gunashekaran. Professor Gunashekaran, over to you.
Thank you, Professor Rod and Professor Ganita Singh, for good introductory remarks on the speaker and as well as on the online lecture series. Indeed, I am pleased to participate in this online lecture series, and the very first day, the launching of the online lecture series. And I congratulate the Association of Microbiologists of India and Academy of Microbiological Sciences of India for the great initiative to bring the online lecture series for the benefit of every microbiologist. I'll make a small presentation about the online lecture series because Professor Kuwad, the chairman of the academy, also asked me to give a glimpses of the lecture series and then follow with my lectures with introduction and with a detailed study on the antimicrobial agents. So may I make a sharing?
Sure, sure, please. Dr. Anil Pangal, you can make a brief of today's program and send it to me so that we can send to the press as well.
Wait a minute.
Take your time. Take your time.
Chat. Share the screen.
ஓபன் பண்ணல ஜானு
Ah.
Yes, here you are.
Is it visible?
Yeah, it's visible.
Thank you. It's the first day of the year. I think I have to congratulate the organizers and wish you all a very happy new year, 2026. You will see a lot of scientific happenings in this new year, 2026. And as Dr. Kuwar and Dr. Amita has already introduced the lecture series and the importance of the lecture series, I will give you the views of the lecture series. In this lecture series, the main purpose or theme, I would say the context is to connect the microbiologist and
भाई तो
share the knowledge.
एक्सपी देने से क्या होता है?
One
एक्सपी
of
दे
the
रहे
is
हैं हमें.
knowledge sharing, and second is networking in research. That is connecting the people. That's why I put the microbiology connect. I think Dr. Kuhad has
Hello.
already mentioned that is going to be the
Hello.
title. I do not know whether it is finalized, the online
Hello.
lecture series title,
मेरी
Microbiology
आवाज नहीं आ
Connect.
रही ना.
And the third
हेलो
point
हेलो
is the capacity building.
एक्सपी देने से
So
क्या
we
होता है?
give the techniques and
गुड मॉर्निंग.
the contemporary
गुड मॉर्निंग.
lectures so that the
गुड
youngsters
मॉर्निंग. गुड मॉर्निंग.
work on in the
गुड मॉर्निंग.
microbiology
नहीं आ रही ना?
area and have a capacity
ये
building
एक्सपी देने से क्या होता है ये बताओ.
microbial sciences.
हाँ दे
Therefore,
रहे हैं
the
लोग.
lecture series invite the experts to discuss and share the advanced research and the cutting edge technology and translational research and innovation in microbial sciences. I'm the first person to start with this
क्या
lecture
दिखा रहे हैं?
series. Thanks to Professor Kuwar and Professor Namita for giving me the opportunity
मतलब
as
मेरे
the
पास जीरो
first
एक्सपी है.
person
लोग
to
देख.
give a lecture in this lecture series. And also, this lecture series will connect the researchers, microbiologists, clinicians, industry R&D scientists, and the students. We are going to have lectures from microbiologists from various disciplines and also from industry persons, and so that the students get benefit of the lectures from the experts. And the theme may include all areas of microbial sciences. It's not limited to what I mentioned here: agriculture, medical, clinical, industrial, environmental, microbiology, or nutrient cycle, public health issues, to contemporary microbiologists. There is advances in microbial sciences, microbial biotechnology, bioinformatics, genomics, proteomics, microbiomes, artificial intelligence, climate change, and system and synthetic biology. So these are the areas to make. And out of these lectures, say online lecture series, what's the expected outcome? So the students and scientists will get improved exposure to microbial sciences and have an academic excellence.
ये मेरे को लोगों को समझ
And
नहीं.
secondly,
ग्रुप में
the
लिख रहे हैं
research
डिस्कनेक्ट हो रहा है. यूट्यूब
idea
पे एक्चुअली
exchange
डिस्कनेक्ट कैसे हो
happens
सकता है?
with this lecture series. And the people who are listening to lectures and they're delivering the lectures
Unable to
make
join. Unable to join.
collaborations and make a networking so that their research can increase the productivity. And definitely this will improve the capacity building in advanced microbiology for the students. So the students will be mostly benefited by getting an exposure to the technology and also the scientists who are working in the area of advanced microbiology sciences. And also, we concerned with the public issues and what's happening due to the microbial threats, and we find out the solutions. So we also focus on the awareness of microbial threats and solutions. And we also give a tip for startup cultures and also connect with the industry so that microbe-based startup industries can come up, and those who are youngsters, they can make a startup, or even the experienced people can also make a startup by getting in the lectures and connected with the industry personally. So these are the general idea with which this online series, lecture series is starting today. And I think at the end of the year, we will really evaluate what's the outcome of this the lecture series, how many participants and how things are received by the scientists and the students. Then appropriately we take a action based on the input from various participants in this lecture series. So in this lecture series, my lecture will be on the mitogenomic
Beautiful
approach for the
disturbance,
development of
like you.
antimicrobial peptides. So this is one part. I will give general introduction about why the antimicrobial peptides need to be developed, and also some of the techniques what we use in our laboratory to develop antimicrobial using metagenomics, and what's the relevance of this the antimicrobial antibiotic developments in this
हम्म कोई
this time.
किसी और के बोलते होगी तो आवाज नहीं आ रही ना.
So our major research area at MKU, various disciplines as Professor Kuwad mentioned, since 80s we worked on Pseudomonas nobilis, and we had experience in cloning and expression of variety of industrial enzymes, and also we started a new program on genomics in 2000, and we focus on microbial genomics area where we develop markers for a variety of microorganisms and study the interaction through the gene expression studies, and also we try to understand the global gene expression at the level of root colonization and the plant growth promoting bacteria. So the other area we focus on is the metagenomics, and we identify the variety of peptides and enzymes from the metagenomics originated from the animal skin metagenome or marine metagenome or oral microbiomes and termitarium metagenome. In this, I am going to focus only on the marine metagenomic, where we identify one of the peptide and I give you a detailed study, and also I touch upon a little on
भाई
the
एक्सपी भेज रहे हैं
antimicrobial.
25 एक्सपी.
So why we are
मेरे पास
looking at
XB रहा भेज नहीं
looking
सकता.
for the
अब किसी के पास वो भेजा तो
antimicrobial
मुझे क्या मिलेगा?
peptides or antimicrobial agents?
हाँ.
And the antimicrobial resistance is one of the global health and development threats worldwide, and it is reported that 1.4 million
ये
global
मेरा
deaths
पहला
will
लाइव
occur
लेक्चर है इस आईडी पे.
entirely due to the antimicrobial resistant
हाँ हाँ आगे बता.
bacterial and microorganisms.
अटेंड कर
In
रहा है.
2021, around 3 lakhs every year death happens in
इस
India in
बात तो
2019.
ईमेल आईडी से
So
होती है ना, या बंदे से
additionally,
होती
you
है?
have at least 2 to 3 times more
ओ,
due to the
लेकिन अभी जिसे
combination
मैं
of the
जिस आईडी से
antimicrobial
स्ट्रीम किया हुआ है ना,
resistant organisms along
पहली बार
with
कर रहा
other
हूँ.
diseases like AIDS and other diseases. In 76 countries, 42% of the third generation cephalosporins, resistant E. coli, methicillin resistant
देखते जाओगे.
Staphylococcus aureus, they
अच्छा,
are major concern.
तो बस जिन्होंने देख
UK
रखे हैं ना उनको
caused E.
एक्सपी ली हुई
coli,
होगी, वो भेज
more
रहे
than
होंगे.
5 cases exhibited reduced susceptibility to the regular antibiotics like ampicillin, cotrimazole, and fluoroquinolones in 2020. This is making a harder effectively to treat these kind of infections. So therefore, economics also says that okay, the antimicrobial agents has a significant economic cost, and World Bank estimates that this could result in $1 trillion additional healthcare costs in 2050 and 3.4 trillion cost GDP losses per year in
वॉइस ठीक आ रही
2030.
है, ये फिर
WHO
अभी एक ने
report
मैसेज किया कि कोई बोल रहा है कॉन्टिन्यूसली।
and global assessment of surveillance and the antimicrobial agents report in 22 also this indicates this one. Coming to Indian context, the antimicrobial resistance is a growing threat to overuse of antibiotic. The Bangalore doctor says it appears in the Times of India newspaper 24th November 2025, just 2 months back. They say that the widespread prevalence of antimicrobial resistance in the country shows that a paper published in the Lancet has revealed that 83% of the Indians carry antibiotic-resistant bacteria. This is only report. I do not know how relevant it is, how accurate it is. Doctors in Bangalore collaborate these findings, saying that they observe daily some degree of antimicrobial resistance in 60 to 70% their patients. So this is a global concern, and also it's a concern in India. Our ICMR report in 2024 also says that it's India's rising threat is anti-antibiotic resistance organisms. Antibiotic losing power, that says in 2024, fluoroquinone, high resistant diarrhea bacteria, typhoid bacteria is 95% greater than resistant. So this is becoming more and more resistant. Third generation
अब चेक
cephalosporin,
करिए।
widely infecting UTI diarrhea, also becoming resistant. So these are some of the data given by the ICR in India that also reported subsequently in Times of India 28th November 2024. Superbug resistant to key everyday antibiotics. Therefore, this discussion on the antimicrobial resistance and the development of novel antibiotics or antimicrobial agents in general is an important area to deal with as microbiologists. So microbiologists should talk on this. Therefore, I have chosen this topic from my experience to discuss this issue in the lecture series. These are the 10 most dangerous antibiotic resistant bacteria listed here. I think all the students might be aware of MRSA, Bacteria, Pseudomonas aeruginosa, Clostridium difficile, Klebsiella, E. coli, relevant to different kind of diseases, Mycobacterium, Neisseria, and Streptococcus pyogenes. These are list of diseases where you found the most dangerous antibiotic resistant bacteria, and sometimes you say that there are few bacteria, we call it as escape pathogens. So Enterococcus, Staphylococcus, Klebsiella, Acetobacter, Pseudomonas, Enterobacter. These are all escape pathogens. They are a greater number in the antibiotic resistance. Therefore, our Department of Biotechnology, Government of India, also recognizes antimicrobial resistance as a top national priority and have several initiatives to address the challenges of pathogens like bacteria and fungi, developing resistance against existing treatment. So I think you can go to the DBT website to see what are the schemes available to get the funding from the Department of Biotechnology. You can work on the antibiotic resistance in microorganisms. Not only in bacteria, the fungal also very much concerned because bacteria there are several antibiotics you can work on, and fungi there are limited number of antibiotics, but even then the pathogenic fungi of animals and humans are generally filamentous and they contain chitin and polysaccharides, making it a rigid barrier for drug penetration. Even then you have antibiotics available, and the cell membrane contains ergosterol, which influences the efficacy and the risk of drug resistance. And most antifungal agents are fungistatic rather than fungicidal, and therefore continuous application of the fungistatic agents are essential. But infection clearance largely depends on the host response. Therefore, this is a major concern with respect to fungi. The drug resistance in fungi in incidence in diversity in fungal infection are being increased worldwide, and WHO is monitoring the magnitude and public health impact of antifungal, anti-drug resistant fungal infection on the increased side. The fungal infection can be difficult to treat, but due to drug-drug interactions for the patients with other infections, and the emergence of spread of multi-drug resistance and candida, like you have it in Staph aureus in bacteria, you have a multi-drug resistant candida, which the invasive fungal infection is a popular concern, a particular concern. Therefore, the development of the antibiotic and list the pathogens which are listed by the WHO, the fungal priority pathogen list included in the comprehensive review in the fungal infection and drug resistance globally. Therefore, not only bacteria and fungal and other microorganisms, viruses, so we should talk about it. And strategies in general, what you see in the laboratories and in the industries also, to the antimicrobial development against antimicrobial resistant organisms is that we have a traditional antibiotics and certain kind of modification you can make it with the existing antibiotics so that the spectrum and the specificity can increase, and the spectrum of antibiotic can be increased and improve the stability of the antibiotics. And also you can develop a new drug class of antibiotic with a unique mechanism of action, and such as targeting membranes, cell division proteins, metabolic pathways which are absent in humans. So particularly active against the pathogens. And also we have the contemporary microbiologist deals about the phage therapy, use of bacteriophages to specifically kill the bacteria, or CRISPR-Cas-based antimicrobial precision targeting the inactivation of the resistant genes and selectively kill the resistant strains. In this also they use phage as a the medium for the delivery of these antimicrobials, or you can use nanomaterials or for the transfer of these antimicrobials. There are detailed study on this. I am not going to detail on that. Similarly, the genomics and artificial intelligence approach is also used to develop a new antimicrobial agents against the antimicrobial antimicrobial resistant organisms. One of the antibiotics is developed by the Institute of Technology of India, MIT, in Cambridge. Yes, one of the antibiotics they developed using the artificial intelligence is a halicin, and this existing antibiotic is, database was screened using the artificial intelligence and making a modification on the existing antibiotic and made it available for a new antibiotic which is called HAL 9000. And this involves a lot of database survey or screening and identifying the antibiotics and increasing the spectrum of antibiotics. So this is several kind of approaches available, and what we are looking for is the peptides instead of antibiotics, and we are calling it as therapeutic peptides or antimicrobial peptides. This can be derived from various sources. Yes, peptides are available everywhere. All living organisms have the peptides. So the plants, animal, and marine, and human, and peptides can be identified through the recombinant libraries and or chemical libraries. So in this case, we started using a newer approach called culture-independent metagenomic approach to uncover the novel antimicrobial genes and gene cluster involved in these peptides. So some of the reported one are having the bioactivities. They are named as teragens and vialogens, indirubin, carbamazine have been reported from the soil metagenome. And our metagenomic studies revolves around the skin metagenome, oral metagenome, and we work on marine metagenome, dermatology metagenome, and so on and so forth. So we found that some of the metagenomes, I think we can skip this. So I will give a brief introduction about the genomics so that the students get the benefit of it before going to the metagenome, and talk about the genomics. Genomics is the study of genome sequences of all organisms, maybe plants, animals, viruses, including humans, and their organelles, their gene organization, mutations, and consequences. The study of information flow through and exploiting the genome databases for identification, characterization, and development of the new organism or products for the human welfare. So here it comes, the drug development area also comes in this. So the different areas of genomics, structural genomics, which talks about the structural of the macromolecules, and you have the functional genomics, the activity of these macromolecules, and you have a comparative genomics, the genome-wide comparison or protein-based database comparisons, and so on. The organism to organism, strain to strain comparison, this kind of comparative genomics can deal with in the genomics area. And the application of genomics has a wide applications in the drug development, disease diagnosis, molecular medicine, and the energy sources, environmental application, risk assessment, and bioarchaeology, anthropology, human evolutions, human migrations, and DNA forensic, DNA typing, and plant health improvement, livestock health breeding, industrial bioprocessing, and several others. So genomic and genomic tools and techniques have a greater potential for sustainable development, increasing availability of food, feed, and raw materials, improving the animal and human health, including the environment. So this one of the area in the genomics is the metagenomics. Metagenomics is otherwise called environmental genomics. So environmental genomics is known as a metagenomic, uses the DNA sequences, sequencing the DNA using the radio technique, and DNA is isolated from the environmental samples without culturing the organisms, allowing the researchers to study the microbial diversity within the specific ecosystem and potentially identify biomolecules by assessing the genetic information of the uncultured organism. So the key aspect of metagenomic study, one of them is microbial diversity, the presence of microorganisms in the environmental sample. Some of them may not be cultivable and some of them may be culturable. So the direct analysis of the environmental sample, high throughput sequencing, functional metagenomics, bioinformatic analysis is one of the area, several of the area in the microbial study, microbial diversity, and the application of metagenomics in biomolecule discovery or enzyme discovery, antimicrobial discovery, for biofuel production, bioremediation, probiotic development. These are some of the area which has a wide application through the metagenomics. And to give the introduction about the metagenomics, it's a new field of the genomics and of molecular biology. And you can very well know that whatever we cultured in our laboratory is less than 1% of existing what is in the environmental sample. So suppose if you take a soil sample and we estimate the number of microorganisms by staining technique and simultaneously compare with the number of organisms identified by the dilution and culturing technique, counting the number of organisms on the plates, and you will see the huge variations. The total bacterial count, one of the studies says that 10 to the power 10 by fluorescent microscopy, but what you could cultivate, the colony forming unit is only 10 to the power 6. So which means that all the bacteria which is existing in the environment, you cannot culture them, but you can get the genes out of those uncultured organisms through the metagenomic studies. And the metagenomic studies is, as mentioned, it's a paradigm shift in the microbial diversity. And there are people who started the RNA genes are conserved and provide phylogenetic relationship. I think the students may know about the Carl Booth, which he mentioned in 1997, where you can take it RNA genes as a gene sequences, you can take the organisms. And then Norbert Peace and his colleague performed phylogenetic analysis of the microbial communities directly isolated from the environmental sample using the PCR technique, and they can they analyze their microbial diversity in the samples. And Joe Adelsman is the one who designed in the term metagenomics in 1981. There are other scientists who followed with applying genomic techniques without isolating the organisms in that. So we are using the same PCR technique and automated sequencing techniques to reveal the microbial diversity, as well as identifying the genes from the metagenomic library. So the general tool, the flow chart for RNA analysis is that just isolate the DNA from the environmental sample. So I will not go into the details, because you go to the manuals, it is available for DNA isolation. It's not the time to explain how DNA is isolated. So you isolate the DNA from the metagenomic sample and amplify with the adapters or primers of RNA genes, and construct the gene library and PCR amplify it with the universal primer, and differentiate the clones by the different methods, ARDRA, DEG, or DGG analysis, sequence them, differentiate the clone, and confirm, validate with the camera check in the tools. And finally, you analyze the phylogenetic analysis of organisms available in the environmental sample. So this is a standard method available. This is the one which share the DNA and then clone it in the suitable organisms, like in E. coli, and recombinant library can be screened, and finally use the Sanger's method for sequencing them. And one can also use the advanced technique as 454 sequencing, and we use the 454 sequencing for the metagenomic sequencing, sharing the genomic DNA, ligating to the adapter, amplify, and using the denature the DNA and pass through the suppressed beads, and then in situ amplification of the DNA, and pyrosequencing the individual beads as colonies and polypeptides. So this automated sequencing system and the machines is favored the easy sequencing of the variety of the clones identified through the metagenomic studies. So accidentally, we were working in a Nimitli project in the DBT with the association of several institutes in the nationwide, chaired by the CLRI Chennai, headed by Professor T. Ramasamy. And in that group, I happened to work on the degradation of the skin. And as an expansion of that, we started working on the same samples to identify the metagenomic DNA of the goat skin. And we found that some of the choline-like peptides are present in the environmental samples. So this is the assumption was made because the DNA, when the skin degradation happens, there is a cycle of events happens. There is the change of the microbial community from time to time. Therefore, one organism gets removed by the other organisms, and the second organism is removed by a third set of organisms. So this kind of happening may be due to the kind of the peptides which can lyse the related and unrelated organisms in the same communities. Therefore, we identify a putative choline-like structures and through the metagenomic studies, and we found deletion consent and find out the peptide sequence, and we make a deletions of these peptides, So 34 amino acid containing peptides, there's a huge antimicrobial activity, and we know how is that the 34 amino acid is essential, but still we can delete at least 11 amino acids from that. Still, it can show the activity. So within the 32, 33 amino acid will be sufficient for making this peptide. A small peptide can be active against a number of organisms. So the variety of organisms, I would say the indicator bacterium like the bacillus, lactobacillus, like all gram-positive organisms, and you have E. coli, Pseudomonas, Stria, these are gram-negative organisms. There are some constructs we could develop. These peptides can make the gram-positive as well as gram-negative organisms get killed. No, so that we since it is a small peptide, we also synthesize in vitro the synthetic peptides. That peptides also we compared with the the peptides we got it through the metagenomes. It correlated very well, so whatever we got the peptide one, we could use it for the deletion the control of the variety of microorganisms. So having this idea, what we thought, okay, control of bacteria, several antibiotics are available, but there is a limitation for the antibiotics, antimicrobials for treatment of fungal pathogens. Therefore, we focused on fungal the pathogens, and we could identify some peptides which can be active against the fungi. So this I will give you some detailed work on how we did it, and then until we identify the nature of the peptide. So I think due to the limitation of the time, I will go through quickly, and finally we will have a, share this one PowerPoint so that any people can have an idea about what it is present in this one. And we took the samples from Andaman Sea, and this is in collaboration with the Pondicherry University, Professor Jayachandran, and we worked together in this area through a joint project. And we constructed a metagenomic library as we mentioned earlier. And this time what happens is that we screened these clones for antifungal activity instead of antibacterial activity. The test organisms we used is the Candida albicans, and the functional the clone and expressions were done in E. coli, and variety of screening methods have been used. Finally, we use a plate technique, the multiple technique, multiple plate technique. I think we examined more than 5 to 6,000 clones, and finally we end up with one clone showing the antifungal activity. And this happened, we sequenced them, and then we identified the ORF, and it was located at 1.6 kilobase fragments, and we PCR it and then confirmed the presence of the genes present in it, and we cloned it and overexpress it, and we call it as a marine metagenomic peptides 1, and this peptide is a small peptide, and it has a 5.4 kilodalton the peptide molecular weight. Then we confirmed through the mass spectro, and also we try to confirm whether it is against only the unicellular fungi like Candida or yeast, or it can also effective against filamentous fungi. So we compared the antifungal activity against the Candida albicans and Aspergillus niger, and Candida albicans the minimal inhibitory concentrations was 0.44, and minimal inhibitory concentration of the synthetic peptide. So because it is a smaller peptide, 30 amino acids, so this shows a 0.57 micromole. As far as with respect to filamentous fungi, the minimal inhibitory concentration is much higher. It's 4.4, and the synthetic peptide is 4.29, which means that this is more against the unicellular fungi. It can also be effective against the multicellular or filamentous fungi. And then we studied the dose response against the antifungal, against the candida albicans, and shows that minimum concentration of the antibiotic required for the inhibition of this one, and different concentrations were used for the analysis from 1.5 micromoles to maximum, and then the lowest level is 0.34. So within 1.2 micromoles, you can easily eliminate the candida albicans. Then on the plate assay, we try to study the thermostability of these small peptides, whether it can stable at the higher temperatures. We have seen that up to 80 degrees centigrade, this is highly stable. Therefore, different temperature used in this study from 30 to 90, so it shows that it's highly stable. And we try to explore the mechanism of action as well as the wide spectrum of the antifungal activity of this MGCP1. We treated with Aspergillus niger and the spores, because earlier we checked with the filamentous fungi, and we try to treat the spores with these peptides, and we could see the complete degradation of the spores after incubation for 36 hours, similar to the degradation of the mycelia into clumps, the degraded mycelia. So within 36 hours, these peptides can easily degrade the fungal system in vitro. And then other bioinformatics studies, protein structure analysis, it shows that it is well preserved structures with a helix and loop, and the CD spectrum also shows that it is the binding domains, and the sequence of the amino acid shows that you have the positive and net charge more positive. The amino acids are available there, and so this MGCP1 predicted using the ITASR, the amino acid predictor. So you have a lot of helix structures, and then you have a loop structures. This the small peptides is highly affected, and it shows a very highly hydrophobic nature. And the size is 4.6 kilodaltons, roughly 5 kilodaltons when you do it the mass spectrum. And a cationic charge, and you have an alpha helical with a single disulfide bridge, and amphipathic helix with a relatively hydrophilic and hydrophobic acids. And then we try to study the mechanism of actions. So since we are concerned with the fungal system, one of the important property is that the resistance is that the chitin or the polysaccharides around the cell wall of the fungi prevents any drug to move into the cell. Therefore, the antibiotics, antimicrobial agents should pass through this barrier. Therefore, we try to find out whether this has the ability to pass through this cell wall. Then one of the important things is the chitin binding assay. So we try to bind these peptides with the chitin and elute it with a gradient of NaCl, and found out whether this peptide has a binding property. And then you can very well see that when dilutions, you see the protein which eliminated, which is the control. So when you bind it with the chitin, so there is no protein, which means that it bound to the chitin of the cell wall of the protein, cell wall of the fungi. And when you sequence them with various other the peptides isolated from animal origins, then we see that there is a conserved region in the cysteine in the proteins, highly conserved residue peptides shown, but it's not identical to the many of the peptides which is known so far against the fungi. And then to study how it, when it binds to the cell wall, chitin, it could go into the cell through the membrane, crossing the membrane barrier. And there are a variety of mechanisms so that it can pass through that. One is energy dependent mechanism, another is energy independent mechanism. And energy independent mechanism, there are two theories. One is it forms a barrel stave mechanism, so it can bind on the membranes and then disrupt and depolarize the membrane. And the secondly, it can binds on the membrane lipids as a carpet, and then it makes us pores and forms a micelle, and then micelle pass through the cell membrane. And energy dependent mechanisms, it's different ways, and then it could entrap the cell through the energy dependent mechanism, and then pass through the macro optioning like structures and goes inside the cell and gets degraded, releases the antimicrobial peptides inside the cell, thereby degrading or leading to the death of the fungi. And one is energy independent uptake, another is energy dependent uptake, that we wanted to test whether it is energy dependent method or energy independent method. The two simple tests is that, okay, if you reduce the temperature, okay, the energy production level will be low. Or you stop the energy production by using azide, then also you will see that the energy production less. So if you treat these proteins tagged with say closed molecules, FATC, then you can see the presence of the drug or antimicrobial agents inside the cell through the fluorescent microscopy. And we found that in all the cases, whether you keep it at 4 degree or 30 degree or treat with a sodium azide, you see the transport of this protein into the cell, which means that it is energy independent mechanism. Then we try to find out the time kinetic study of this the proteins passing through the cell wall and cell membrane, and we found that within 12 hours most of the proteins get into the cell. This also using the FATC tagged proteins and fluorescent microscopy analysis. And then to find out, substantiate this one using the unicellular fungi, we use the flow cytometry analysis to find out how many of the cells or gets taken up the proteins within this time. We found that within 6 hours, this unicellular fungi, that was one is filamentous fungi, this one is a unicellular fungi, Candida albicans. Within 6 hours, 99% of the proteins goes inside the cell, is an energy independent mechanism. Then, okay, when it goes into the cell, what happens? What is this protein action on the cells? So one of the prediction is the cytotoxicity. So majority of the proteins, they lead to the cytotoxicity in addition to the cell lysis, or through the cytotoxicity leading to the cell lysis, or independent of cell lysis cytotoxicity, lysine the cell membrane, or kind of like the lysozyme and other things. So in this case, we target the cells with the proteins MMGP1 and they harvest the cells and stain with the syntax screens. So this syntax screen normally fluoresces the macromolecules within of the cells once it is dead. So dead cell will give the fluorescence and the live cells will not give the fluorescence. So we do the analysis of filamentous fungi and the albicans. We really confirm the death leading to the mediated through the peptides happens in the cells. So using this syntax staining methods, we show that all the cells are dead. We cannot cultivate and see how many number of cells are died because some of them these are the filamentous fungi, the cellulose fungi. Therefore, we took a staining method to find out how much time it takes to completely eliminate these the cells pathogens. Okay, so within 30 minutes, you can see that. This also we done with the flow cytometry to prove that the cell death occurs in Candida albicans. The flow cytometry analysis show that 99% of the cells dead in 24 hours. So there we show that in 6 hours it goes inside, but the cell death takes about 24 hours to complete loss of the life of these cells. Then to find out what is the nature of the mechanisms which lead to the death of these organisms, these peptides lead to the production of reactive oxidants, and this also we use techniques, dichlorofluorocene diacetyl, the staining technique, and these dyes can be oxidized by reactive oxygen. So suppose if reactive oxygen is produced or hydrogen peroxides are produced inside the cell, the level of hydrogen peroxide production or reactive oxygen presence can convert these non-fluorescent probes into the green fluorescent probe. So these staining techniques really help us to find out whether this protein goes inside the cell and leading to the death due to the production of reactive oxygen. So the general observation through the staining technique show that this happens. See, the assessment of MMG production through the flow cytometry, it shows that within 180 minutes, you see the increase in the production of the reactive oxygen. And then the reactive oxygen is produced, whether it is due to the production of the hydrogen peroxide or presence of hydrogen peroxide inside the cell, because reactive oxygen production leading to the hydrogen peroxide, and this can be estimated by using another technique called DAP, diaminopenicillin stripes, which can lead to the formation of the reddish brown complex when the target cells producing hydrogen peroxide interact with the DAP, leading to the precipitate of reddish brown color. So this also shows that with respect to the cells, it forms a reddish brown color. So which means that with the reactive oxygen produced and through the hydrogen production, so general mechanism known so far is that reactive oxygen can lead to several damages inside the cell. Reactive oxidants can lead to the protein carbonylation, lipid peroxidation, or DNA damage. So these are the major activity, and which means that these lipid peroxidase can damage the macromolecules like DNA and affect the cellular oxygen metabolism, or it can degrade the proteins, and it can also affect the mitochondria through the lipid peroxidation. So all these combination of these effects can lead to the death of the organisms. So a simple 31 amino acid small peptides can easily make changes in the peptides, the fungal system, and this also we confirmed through the assessment of the ROS production through the DNPS assay, and the protein carbonyl content was measured by the DNPH assay, and you have the lipid peroxidation by the Kafka and rhodamin staining technique. So all these technique, this rhodamin it's a technique. It shows that the lipid peroxidation happens in the cells treated with this one peptides. So the mitochondrial functions was also tested with the acridine orange. And the staining of mitochondria with exposed to MGC cone with the acridine orange, and we found that the cell becomes inactive because of the inner depolarization of the membrane. So the mitochondrial treated with 1 micromoles of hydrogen peroxide was used as a control. So all these things shows that the DNA damage in the cell happens, and which can be identified through the photomicrography of in vivo cells induced by MGGB1, and you can also see other staining technique. Finally, prove that this is very effective against both unicellular and filamentous fungi. So as I shown that this unicellular and the filamentous fungi can, if it gets killed by them, it can be effective against the human cells. No, to find out whether it is any effect on the human cells, we started using in vitro assay the hemolytic activity of this protein on the RBCs, human RBCs, and we treat these cells with a different concentration of the MGGB1 and with HRBC for 1 hour, and the hemoglobin release was estimated as a hemolysis by the calorimetric method, and calculated the amount of the peptide required for the inhibition of this one. So we found that the minimal inhibitory concentration for this one is a 4, and if it is 18 in the microgram, there I mentioned about micromoles, this one, and we use MHC for this one 50, the constant one, and the therapeutic index. The therapeutic index is the ratio of MH values over the geometric use of MAC, and a large value indicates a greater antimicrobial activity. So the larger value indicates the antimicrobial activity rather than the cells against the human cells. So this indicates that at a lower concentration, you can eliminate the fungal pathogens, and if you increase the concentration at a higher level, then it may also lead to the toxicity to human RBCs. So finally, the mechanisms, it shows that this antimicrobial peptide was identified through the metagenomic analysis, and try to understand the mechanism of the antimicrobial activity of this peptide with the following steps, and finally we formulated the model showing that this is a kind of peptides which uses these modifications in the cell, binding to the chitin first step, and then the peptide aggregate and directly penetrate through the membrane through energy independent mechanisms, and finally when it goes into the cell, it induces the reactive oxygen species production, and which can lead to the protein carbonylation, DNA damage, membrane degradation, plasma membrane degradation, and finally it leads to the death cell. This is a final model for this peptide. So it is only at the laboratory level. So these are the publications made out of our metagenomic studies. So it's not only on the marine metagenomics, other metagenomic studies also we covered here. More than 23 papers in the journals which are published only on the metagenomics. And the collaborators in the metagenomic studies from our group, these are our graduate students who work on variety of metagenomes, from skin metagenomes to water metagenome and marine metagenome, and then we have a termitory metagenome and so on. We have collaborated with scientists from Pondicherry University and his graduate student. And in the world metagenome, we have a collaboration with Kendall Institute in London, with Peter Murphy, and they are the scientists from there. And all our the projects on metagenomes were supported by the DBT in New Delhi. And the facility is developed under the Center for Advanced Studies, Center for Excellence in Genomic Sciences, Networking Research Center in Biological Sciences, and Integrated Program in Life Sciences at MKU. It helped us to carry out this kind of genomics and metagenomic studies in MKU. So with this, I thank you all for patient listening to these lectures. And this is very lengthy lectures, and that's why I made it as a very quick scan through. And if possible, the slides can be shared through the office of the NAMC to the participants of these lectures, so that they get benefited by listening and complete understanding of this lecture. Thank you.
Okay.
And
Thank you, sir. Thank you, sir. Thank you for a nice presentation. Sir, we received few of the questions from the audience. That question first is from Professor Halami from CFTRI. What are the approaches or should follow to develop new antimicrobial drug that not have any AMR development issue?
well, that's what I mentioned. No, there are the methods already available. One is standard the methods like modification of this existing antibiotic. That's how it is semi-synthetic the antibiotics like ampicillin, cefoxitlin, and all kinds of penicillin derivatives. So that is a modification of antibiotics, and this has a wide stability and improve the spectrum and reduce the toxicity. And you have other class of antibiotics you can use using the new drug which use these methods. You have to understand the pathways of the organism. So first you have antimicrobial resistant pathogens. Then first you understand the pathways of this organism and develop a drug against the pathogenicity, or sometimes it produces a toxin. So you can target the toxin of the pathogen rather than the pathogen itself. So, or the pathogenic principle of the pathogens can be targeted. So develop against this and the pathogens. Or you can develop the antibiotic or antimicrobial agents which inhibit cell division because specific to the cell division of microorganisms which cannot affect our human cells. So recent days, these two techniques are very popular. One is a CRISPR-Cas based technique, and this selectively uses the modern technique like screening of the resistant genes and targeting the precise. I have one article, it's a recent article, we can see it in these, I think I saw in one. I can share it. The NIH recently they found one of the antibiotic through uh, the
CRISPR-Cas.
CRISPR-Cas based antimicrobial, and which actively effective against inactivating genes used in the antimicrobial resistance, the biofilm formation of this pathogen, and this very active against E. coli, Enterobacter faecalis, and Staphylococcus aureus, Clostridium difficile. And I think that paper I can share it. It is not our paper, but I saw recent papers. One through the this method is CRISPR-Cas inactivation of the chromosomal resistant genes, and also the curing of the plasmid through the CRISPR-Cas mechanisms. So this is the kind of the approaches one can use it.
Okay, sir. Then the
And then genomics and AI tools also recently now being used. That's what I mentioned. One of the antibiotic was used the AI tools also early since. This is the NIH scientists who NIH from Cambridge. They have developed this kind of broad spectrum antibiotics.
So then another question, I think that it is clearly mentioned by you, the strategies you can follow. That because another question is what was the idea to examine the goat skin microflora for the discovery of new AMP?
So yeah, this was we were working on the Nimitli project. Yeah, we are working on the Nimitli project to eliminate the chemical-based preservation of the leather, the skins for the leather processing. So as you know very well that in the leather processing, the initial step is the preservation of the skin using the salt. So this salt poses a problem in the environment. Therefore, one of the idea was to replace the salt with biodegradable materials so that it can be used. So there we were involved in identifying the microbiome, the community dynamics during the degradation of the animal skin. So we found that there is a succession of organisms which are present in the different type of environment. Initially, start with the gram-positive organism. I'm telling in majority is gram-positive organism, and then gram-negative organism, then multiple of several organisms, and other fungal system later get into. So in this process, what is the mechanism involved is that the microbial dynamics is through the degradation of the communities. So this community is overtaken by the other community, not by the chemical means. It is through the production of some enzymes or some macromolecule which eliminate that. All students may know that the presence of holines and lysozymes, this kind of proteins which can degrade the bacteria. So that was an idea. Okay, it should be some new holines which we may find it through the metagenomics screening. So that's how one. And for the project we were working on the degradation dynamic analysis, but it is another PhD thesis. We started working on the metagenomics, identifying the novel peptides through the samples we had it in the skin dynamics. We also worked on the human skin. That I will not make a presentation. That's a small work, and how the skin bacteria changes with respect to time. So this idea is that the microbial dynamics changes due to the degradation of the community by another community through the production of the peptides. So that was in principle, we got an idea.
Sir, another is related with the fungi. How challenging is AMR in fungi than the bacteria? Selective
Yeah,
action of AMP against fungi and not to the human host.
yeah. That's why these smaller peptides, which can be used for the fungal treatment, uh, they, they, they are they are effective against the fungal system at low concentrations, and it can be eliminated by the degradation. So it is effective against the microorganisms, not against the human cells. That's why the RBC study, HRBC study shows that you require the high concentration of this one to give a toxicity to the human cells. So it is effective against the the microorganisms rather than the higher organisms. So one of the problems here is that the stability of these proteins, how long it is going to be stable and how long it has to be treated. So these are some clinical studies you have to carry out with a collaboration with medical scientists, so that this can be done. So yeah, as of now, at the lab level, we've shown that this is effective against both filamentous and unicellular fungi. Taken to the clinical level, it requires much more time than this.
Yeah, that is very correct, Dr. Gurushekar. Thank you very much for the very informative talk. And that is what, I also wanted to say, especially this bacteriocins have been very helpful to,
Yeah,
take care of this havoc created by antibiotics. So,
yeah,
of course, fungal has to be seen in a later date.
yeah, that's correct.
Okay. Thank you, sir. Thank
Thank
you.
you.
May I request now the Professor Ruplal, our past president, AMI, for the closing remarks. Professor Ruplal, over to you, please, sir.
Thank you, Namta. Before I really conclude, I would like to congratulate and compliment AMSC and AMI for jointly launching this online lecture series, which, as mentioned by Professor Kohar and others, will go a long way in propagating microbiology, particularly among young faculty members and students in the light of recent developments that are happening in this particular field. Today's lecture on metagenomic approaches for development of novel antimicrobial peptides by Professor Gunashekhan very clearly highlighted. In fact, one of the major problems that is not only being encountered in India, but also world over, he clearly mentioned in his presentation that this is a very serious problem, uh, uh, and there could be large number of solutions, including looking for new antibacterial or antibiotics, or look modifying the existing antibiotics, then looking for chemical molecules, developing vaccines, and so on and so forth. But out of all these, one of the easiest approach could be looking for metagenomic tools to resolve this particular issue. He emphasized repeatedly that this is a very serious problem. In fact, our Prime Minister last week he mentioned about antimicrobial resistance as a very serious threat in India in his Man Ki Baat. So it is a very timely and nicely elaborated lecture by Professor Gunashekhan, who not only mentioned the problem but also looked into how it can be really resolved by looking into metagenomics and cloning these smaller peptides, particularly in the marine environment from where or the niche that has not been explored, particularly in India. So going for simple cloning and then actually expressing the gene and then looking for simple peptide, going for toxicity and evaluation and mechanism of action. Probably Professor Gonshekar, it's a work of many, many years and of many collaborators. So it's a really wonderfully elaborated example that can guide our young faculty members and students how to really start from scratch and go for actual application or to take it to the logical conclusion. Your experience and also the approach that you have followed probably will become an example for young students to follow and conduct this particular research in this exciting field of metagenomics. Thank you very much for this space time and presenting this your work in such a elaborate manner. Also, would like to thank Professor Archie Kohar and for his vision and also initiating this particular online lecture series, which is very important. And I hope it will turn out to be really very fruitful exercise for MI in the years to come. And thank you, Namita, for really convening it wonderfully. And all our participants who could really make it, I'm very sure this elaborate lecture will benefit you all. Thank you very much.
Now I request Dr. Anil Panghal, our Joint Secretary, for the vote of thanks. Dr. Anil Panghal.
Thank you, ma'am. Thanks, Professor Sridhanasan sir. On the behalf of Academy of Microbiological Sciences, AMST and Association of Microbiologists of India, we extend our heartfelt thanks to this insightful and inspiring lecture. Your depth of knowledge drawn from the distinguished career as former senior professor of microbiology at Madurai University and former vice chancellor of Trivandrum University and VIT Bhopal University and tremendously. The clarity with which you explained the metagenomic strategies, the practical examples you shared, and your perspectives on translating discoveries into therapeutic lead provide invaluable guidance to the researchers, students, and practitioners. Especially appreciate the time you took to address the questions and engage the attendees. The thoughtful responses spark the meaningful discussions and will undoubtedly stimulate further research and collaboration in this vital area. Thank you once again for your graciously sharing your expertise and contributing so significantly to the success of this inaugural lecture in the MSc and AMI lecture series 2026. And we also thanks to all the participants who attended this lecture series from all the corners of India, and without whom this lecture series will not be successful even. We request all of them that we should share these links when we are sharing these links to the you people, with other colleagues also, and students and other PhD scholars. Thanks to all of you.
Yeah, Namita, just a minute before we disperse. I take this opportunity to thank the President of AMI, General Secretary of AMI, and the whole Executive Council of AMI, as well as the Executive Council of Academy, and all past presidents who have participated in this program, because I could see there are more than 10 past presidents who have participated, naming Professor Ruplal, Professor Panda, Professor Yogendra Singh, Professor Judeja, Professor Sunil Papi, and Professor Gunashekhar himself, and many others, as well as our other colleagues who could spare time to make this beginning of this lecture series a success. I'm really grateful to each one of you that you could really spare time, because this is I think very cold weather, as well as being Sunday. But now we have, say, just taken a leap, and as Professor Ruplal has mentioned, we will certainly strengthen this program, as well as this will mark a really remarkable, say, kind of series in the time to come. And in this series, I'm happy to inform each one of you, those who are, say, present here, that our second speaker has also agreed, Professor Amulya Panda, because he has got professorship and he has joined our community as professor. Professor Amulya Panda will be delivering his lecture on 18th of January, same time at 12 noon. And I request each one of you to join.
Thank you.
as well as
Great, great.
spread this message in advance. Of course, officially we'll be circulating the whole schedule as well as the link. But those who are present here, please say note down in your say the mobile diary so that you don't miss it, and advise your students as you have seen, because today's lecture, not only I'm saying that, but I think I could see the comments from the participants that one thing is very clear, that Professor Gunasekharan's lecture was very, very informative. And that, I think, a good success of today's launch of, say, this, say, lecture series. I once again wish all of you a very, very happy, prosperous, and healthy new year. And say, all of my colleagues, they bring a lot of laurels, say, in their career. And lot of thanks to each one of you. Thank you very much. I
Thank you,
need
sir.
your support. I seek your advice so that we can move further. Thank you very much.
Thank you very
Thank
much.
you. Thank you very much.
Thank you, Dr.
Happy
Kuhat,
New Year
founder
to
of
all.
Thank
EMI.
you. Thank you very much.
Happy New Year to all.
Thank you.
Thank you.
Happy New Year to
Thank
you.
you, sir.
Rajesh.
Happy New Year to all the organizers.
Namaskar, Rajesh.
Namaskar, sir. Namaskar.
Very good. Inshallah.
Namaskar. Thank you all of
So nice of you.
organizers.
Thank you, Professor Gunasekharan ji.
Thank you, Professor
So
Kuhan.
nice. So nice.
Dr. Mita, everything's
Now
fine.
that
Namaskar.
meeting may close.
Yes, you can.
Thank you very much. Thanks.
Thank you
Namaskar,
very much.
Kabir ji.
Professor Gunasekaran.
Thank you, Sunil. Sunil Kabir.
Yeah, now I think
See you on
Dudeja,
see you on
we
18th
will
of,
be meeting each other.
I'm seeing Dudeja.
18th of January.
Yes, wonderful, wonderful. Really, really very good.
Thank you, thank you. Thank you.
ये ऊपर